Welcome, everybody. We have now made it to the fourth unit. So this chapter, nervous tissue, chapter 11 in your textbook, is going to be the first chapter that's going to be covered on the final exam. We'll talk more about that later. Okay, but if you're watching it the week that I post it, keep in mind that this will not be covered on exam number 3. So finally, we get around to the nervous system. We have covered not every other system, but we've covered a lot of the major systems of the body. But we haven't said much about the nervous system that coordinates much of what we see in the other systems. All right, so a lot of this is going to be kind of laying out categories and getting us organized. So I would suggest that you might want to watch this more than once to go back over this material and make sure you thoroughly understand these different groupings or categories. Okay, so essentially, if you take the entire nervous system, you can break it into 2 large pieces. You have what's referred to as the CNS, or central nervous system, which consists of the brain and the spinal cord. Okay, so remember, these are in the dorsal body cavities. And then you have the PNS, or peripheral nervous system. These are the cranial nerves. We'll see those, they are associated with the brain. The spinal nerves associated with the spinal cord, and the ganglia that house the soma, or cell bodies of neurons outside the central nervous system. Okay, now what we're doing here is I am subdividing the peripheral nervous system. So I'm not talking about the brain and the spinal cord right now, I'm just looking at peripheral nervous system. Okay, within the peripheral nervous system, in terms of how they function, we've got 2 divisions. So the sensory portion, or what is also known as afferent, includes the receptors and the nerve fibers that carry information to or toward the central nervous system. So if you look at this visual, everything that is going to be gathering information and sending it into the central nervous system is considered sensory or afferent. This includes the somatic and visceral sensory systems that we'll talk about, as well as the special sense organs. Okay, so more on that in a little while. Next, we have the motor or efferent portion. Now, keeping afferent and efferent separate can be difficult. We've seen these terms before. If you go back to the lymphatic system, when we talked about afferent and efferent vessels. So the way I remember it is that efferent starts with the letter E, just like the word exit. So if it is efferent, it is going to exit. All right. So these are going to be exiting the central nervous system. These include nerve fibers that carry action potentials or impulses away from the central nervous system. And we will be able to divide this further into somatic and autonomic nervous systems. Okay, so this I think is really useful, and I would urge you to write this out maybe in your own way, or even to print this and have a copy of it next to you while you're working through this lecture. So remember, we're just talking here about the peripheral nervous system, and it divides into motor and sensory. And then we said within motor, we've got somatic and autonomic. And within sensory, we've got somatic and visceral. Okay, so if you notice there's somatic, visceral, somatic, autonomic. Okay, so that's where those come from. All right, so we're looking now at the sensory portion. So we're going here. Okay, so we said we've got a peripheral nervous system, then we're dividing to sensory. So that's everything that's coming in the central nervous system. Now we're going to look at somatic versus visceral. Okay. So the information that is being collected, right, everything that's going to be heading into the central nervous system starts with sensory receptors. So there has to be something that responds to a stimulus. So sensory receptors that are considered somatic are found in your skin, your fascia, the joints, skeletal muscle. Okay, so all of this is collecting information about what's going on in the surface of the skin, areas where there's pressure and movement. Okay, so it tells you about where your body is in space and how your body is interacting with the environment outside of the body. Okay, so these kinds of receptors include touch, so that would be a light pressure receptor, pain receptors, pressure, which is basically a heavier touch or is going to penetrate deeper into the tissues. Temperature receptors, so there are receptors for both heat and cold. All of the special senses that we'll be talking about, so that's like visual, smell, and all of that. Okay, and so these receptors tend to be involved more in voluntary aspects of the body, things that you move, information that you collect yourself. Okay, and so it's going to feed back to more of the voluntary skeletal. We'll get there in a minute. Okay, let's go down here to visceral. You know that term visceral, it means guts or organs. So the visceral sensory receptors are collecting information from inside the body, from the viscera. Okay, so these can include temperature. So if you're running a fever internally, you can detect that. And stretch receptors. So stretch receptors are really important internally because they help you to know, for example, when it's time to evacuate your bowels or evacuate your bladder, things like that. Okay, let you know when you're full. So there are stretch receptors in the wall of your stomach that help you to know when you're full. Okay, and a lot of these are going to be tied back with smooth muscles and involuntary systems. All right, so let's go back again. Remember, we followed up sensory. Now we're going to follow those divisions for motor, somatic motor and autonomic motor. Okay, somatic motor, remember that motor is output from the central nervous system. Okay, so this is what is exiting the central nervous system. So somatic motor division involves the voluntary control of skeletal muscle. Okay, so you want to reach up and scratch your nose, you're going to be sending signals to skeletal muscle, which leads to muscle contractions and allows you to scratch your nose. The autonomic motor division is going to tie in with involuntary muscles, smooth muscle and cardiac muscle. And later, we don't get into it too much, but you can subdivide autonomic into parasympathetic and sympathetic. So sympathetic is the fight or flight response, and we talked about with hormones, and parasympathetic is often referred to as rest and digest. So these are more basic maintenance, and metabolism falls into that. All right, so again, maybe go back to that flow chart. Let's do that quickly. Okay, so we've talked now about peripheral nervous system as its sensory portion and its motor portion. We looked at somatic sensory. We looked at visceral sensory. Then we looked at somatic motor and autonomic motor. Okay, so I would keep this on hand because these terms are going to keep coming up and they can get very confusing. Okay, so there we go through motor. All right, now let's move on and talk about neuroglia. So if you throw your mind back to when we were learning histology, you will remember that when we looked at the nervous tissue, I told you we only have two different types of cells. We have neurons and everything else. And all the everything else cells, I said we would call glial cells or neuroglia. Okay, now we're going to look and see what are these different cells. So we're going to look at categories of neuroglia and start by looking at the cells in the central nervous system. So remember, this is the brain and the spinal cord. So these are additional cell types that you find there outside of just the neurons. Okay, first, astrocytes. You see it here in this image. So think astro, that makes you think of stars. So you can imagine this has got sort of central body and these little pieces shooting out like a star. And what do the astrocytes do? Well, they have multiple roles, but first and foremost is they establish what's called the blood-brain barrier. Okay, it's getting windy out here. Okay, the blood-brain barrier, if you see down here, is going to be a layer of tissue from the astrocytes that surround capillaries. And what this does is it gets to part 2 here, which is regulating molecules entering the interstitial space. So remember, the interstitial space is the area outside the circulatory system. Okay, they're also going to control what enters the capillaries. So a blood-brain barrier is a way of strongly regulating which molecules enter and exit the interstitial tissue. So in this case, it's the brain and the spinal cord. So this is really quite important because it means if you give somebody medication to treat certain conditions, it cannot necessarily enter into the brain and spinal cord because this blood-brain barrier makes it quite difficult to get chemicals, molecules into those tissues. All right. So you can just see this one astrocyte has laid down several of these little feet-like structures on the capillary. But as we'll see a little bit later, many astrocytes will encircle and put these little feet-like structures on the capillary, so the capillary is completely enclosed. Okay, and you can see that it also attaches to surrounding structures. So that helps to give us the third function, which is to strengthen the central nervous system. Central nervous system, these tissues are very, very soft. Okay, there's not a lot of protein fibers or anything like that running through the central nervous system. So if you were to do an autopsy, for example, and you were looking at the brain, you'd find that it just is very, very soft. Okay, and then the fourth one we won't really talk about, but astrocytes help the neurons to develop, and they can aid in neuron repair. So here's our neuron over here, and you can see it's touching the neuron. We're going to focus on these first 3 functions of the astrocyte. All right, over here on the right-hand side, we see a row of ependymal cells. And the ependymal cells line openings called ventricles and tubes called central canals that run through the central nervous system. Okay, so within your brain, you have 4 of these spaces, ventricles, and between the ventricles are canals that attach them. And all of these spaces are filled with fluid. It's called cerebrospinal fluid. You can see it right here, often abbreviated as CSF. And that fluid is going to be produced by the ependymal cells. So I want you to imagine you have an opening lined with these cells, and basically what they do is they allow water, primarily water, to leak into this space, and they also control the chemistry of that liquid. Okay, more on cerebrospinal fluid in a little while, but that is made by the ependymal cells. Okay, third are microglia. That's this guy right here. Remember, we're still in the central nervous system. So microglia, these are phagocytic immune cells. Because the blood-brain barrier makes it quite difficult for white blood cells to move in and out of the brain. So you have these specific phagocytic cells that move around within the central nervous system. Then on the right here we see, this is a long one, oligodendrocyte. Okay, so here is an oligodendrocyte. There's the nucleus. That's where all the important organelles are. And then kind of like the astrocyte, it has these branches that extend from it. But it's different. It doesn't end in a little foot-like structure. Instead, it wraps around axons. Okay, so you can see it sort of reaches out and then deposits a layer of what's called myelin. Now, myelin is a fatty material, so it's primarily lipid, okay, and it surrounds axons, and this myelin sheath is going to help the action potential to travel faster down the length of the axon. Okay. All right, so here we see, this is a nice image because it shows all 4 of these different examples of glial cells. So in blue, you see the really big neurons. There's the soma with the cell body, and here's a process. Okay, we see an opening into the central canal lined by ependymal cells that make cerebrospinal fluid. We see the astrocyte cells that are surrounding, excuse me, this is a capillary. Okay, so establishing the blood-brain barrier. Oh, I'm sorry, there's the capillary over here. You see how it's completely surrounded? Okay, so here's a capillary that has been surrounded by the astrocytes and is now has a blood-brain barrier. And you can see also the astrocyte forming connections between structures to strengthen the central nervous system. And then we also have our microglial cell here that's moving around and destroying and consuming cells that don't belong there. And then finally, we have our oligodendrocytes here, 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 and they are laying this fatty material called myelin onto the axons. All right? All right, now for the peripheral nervous system, remember this is outside of the brain, outside of the spinal cord. We only are going to identify 2 cells that are neuroglial cells. Okay, the first are called satellite cells. Okay, and a satellite cell is found in a structure called a ganglia. A ganglia is an area along the nerve that appears swollen. Okay, so nerves themselves are quite thin, but you can certainly see them in a dissection. The appearance of a nerve is kind of like waxed dental floss. It's usually white in appearance and it's shiny. And that is because of the myelin that's been deposited on that axon. So waxed dental floss is a good analogy because wax is also a lipid. Okay, but if you follow these nerves, you're going to find areas where there are swellings. And if you look really carefully at this image here, you're going to see a swollen area, and that is called a ganglia. So if you go inside the ganglia, what do you find in a ganglia? Okay, well, here's your myelinated axons, right? They're inside and they're traveling together as a nerve. But in the ganglia, what you find are the soma or cell bodies. And remember, this is the part of the neuron where we find the nucleus and the other organelles. So it is absolutely essential that the soma be taken care of, be well, basically well maintained, because the neuron, the entire neuron can die if the soma is damaged. And the neuron, remember, can be very long. It can be something like 3 feet long. And as we'll see, it is possible to repair a myelinated axon that gets damaged. But if you destroy the soma, you can't regrow or repair that neuron. So how do we keep that soma safe? Well, here it is. It's outside of the dorsal body cavities, which makes it kind of more likely that it could get injured, right? It's not protected by a skull. It's not protected by the vertebra. Instead, it's traveling through the body where it's more likely to get injured or damaged. So these satellite cells that you see, they look kind of pink and puffy here, surround, care for, and protect the soma or cell bodies within the neurons. Okay, so if you're in satellite, remember a satellite is around the Earth. So think of these satellite cells as being around the soma and caring for the soma. Okay, now the last type of neuroglia, this makes 6 different cells we've learned, is called a Schwann cell. And a Schwann cell is a lot like the oligodendrocyte cells of the central nervous system, because the Schwann cells put myelin on the axons. However, the way they do it is different. Okay, first let's consider myelination. So I told you that myelin is a lipid. It's actually a mixture of lipids and proteins, okay? And they surround and insulate an axon. And what this insulation does is when an action potential travels along the axon, instead of, I don't want to get too technical, and I can't really draw for you, but basically, I want you to imagine that you have an area where you only have small exposure of the axon. The rest of it is coated in this sort of plastic material. So an action potential will jump from one exposed area to the next exposed area instead of passing down the length of the axon. So the upshot, what you really need to know, is that the action potential travels faster, okay? And it uses less energy. Less ATP is necessary to reset that axon. Remember repolarization and depolarization that we talked about with that with the heart? It's the same idea. When an action potential travels down an axon, it requires ATP to reset. So when an axon is myelinated, the action potential moves faster and less energy is required. Okay? All right, so if you're a Schwann cell, how do you put myelin on? Well, the Schwann cell, unlike the oligodendrocyte, does not have multiple extensions of the cell that reach out and wrap around the axon. Instead, one cell, this in pale blue here, this is one cell, rolls itself repeatedly around one location of one axon. Okay, so imagine you're making like a jelly cake where you sort of have a flat cake and you put jelly inside it and then you roll it up tight. That's a jelly roll. I don't know if you guys know what those are or not, but imagine something like that. But you're just rolling the cell, the Schwann cell, around the axon. Okay. Now, what that does is it creates areas that have no myelin. So see right here in yellow, here and here, that is the place where the axon is not myelinated. So this gap, this area where there's no myelin, is called the myelin sheath gap. And it's these gaps that allow the action potential to travel faster along the length of an axon. Okay, so you can see what it looks like here. So this is an axon, and each of these is an individual Schwann cell. Okay, so this is a myelinated neuron of the peripheral nervous system because we recognize that those are Schwann cells. And the only way you get myelination of the peripheral nervous system is with these Schwann cells. Okay, this is a different neuron. Okay, this is an unmyelinated version. And with that, it's going to take longer for the action potential to travel down the axon. Okay, so here's sort of a comparison. Above, we see myelination in the central nervous system with the oligodendrocyte cells. See how they reach out and they myelinate in multiple locations on multiple neurons. Here, we see myelination of an axon of the peripheral nervous system, and each one of these is an individual Schwann cell. Okay, so there's only 1, 2, 3, 4 oligodendrocytes in the top, and then there's 1, 2, 3, 4, 5, 6, 7 Schwann cells in the bottom. Okay, moving on. Let's look at, this is mostly review, but we're going to look at the anatomy of a neuron because I covered only the most basic type of neuron, and it turns out there's more than one. Okay, so the parts of the neuron, we have the cell body or soma, and that's where all the organelles are. Then we have the dendrites, which are typically very short processes, and the action potentials that travel on dendrites are going this way, toward the soma. Okay, so it can bring in action potentials from multiple directions. And then the axon, which is often called the nerve fiber, and that makes up the bulk of the length of the cell, is going to travel away from the soma, and it's going to carry an impulse to a number of places, not just muscles, but this is one example. So imagine you want to scratch your nose, that action potential travels down the length of an axon to innervate one of your muscles, like say your biceps brachii, so you will flex at the elbow, okay? If not a muscle, it could also be a gland, an organ, could be smooth muscle, could be cardiac muscle. Okay, there's lots of different things that can be what we call innervated, right? That the nerve can go there. Okay, so here's this sort of summarizes. Here's one neuron, follow the axon, and you get to another neuron. So this is a synapse from one neuron to a next. Now, you remember we looked before at a neuromuscular synapse. I showed you that when we were looking at how muscles contract, and we focused on neurotransmitters. Okay, so that's the same idea here. So there's that neuromuscular synapse. So if you zoom in here, that's what we were doing previously. Or that same neuron may synapse with glandular material. And that's going to cause, for example, secretions to be released from those glands. All right, now here are the 4 different types of neurons that you can find in the human body. The one we are most familiar with is this last one here called a multipolar neuron. That's kind of the typical. There are 3 other varieties we're going to look at. The anaxonic, okay, notice it has no axon. So it lacks an axon. It's found in the central nervous system, so brain and spinal cord, and it's very rare. Okay, it's basically a whole bunch of dendrites. All right, second one, bipolar. And this is associated with the special senses, like your sense of smell, for example. It has a little bit different because in this case, the dendrite merges together. So the big difference is, notice these are the dendrites, but instead of multiple dendrites coming in to the soma, the multiple dendrites merge into one to the soma to the axon. Okay, so really the big difference here is the merging of the dendrites, and these are not myelinated. Okay, so we're going to see bipolar neurons. We look at the special senses. Then we get to the pseudo-unipolar. Pseudo means false. Okay, so it's falsely looking like it is a unipolar neuron. So what's weird about it is that the dendrites do not go all the way to the soma. The dendrites merge directly into the axon. Okay, you find the pseudo-unipolar neurons associated with the sensory neurons, so this is not motor. Okay, so this is information traveling into the central nervous system, and these kinds may be myelinated. All right, the last one, and by far the most common, particularly for the central nervous system, because the other ones are the bipolar and pseudo-unipolar we're going to see as part of the peripheral nervous system. But the multipolar are the most common type in the central nervous system, where you have a single axon coming straight off the soma. Okay. The other thing to notice is that all the motor neurons, so remember, motor neurons are part of the peripheral nervous system, all the motor neurons are multipolar. So bipolar, pseudo-unipolar are found in the sensory division of the peripheral nervous system. The anaxonic neuron is found in the central nervous system. Multipolar neurons found in all of the motor neurons, and by far the most common of the central nervous system. All right, now we're going to look at the functional classification of neurons. So we're thinking about how neurons work. So the neurons that are sensory, making them afferent, that are part of the peripheral nervous system. So these are neurons that carry information into the central nervous system. Okay, they are most commonly the pseudo-unipolar. Okay, so where are we going to find them? They are coming as exteroceptors. So an exteroceptor is a type of neuron that's collecting information from outside the body. So this could be traveling from the eyes, from the nose, from the middle ear. Okay, so they're giving you information about the world outside. So extero, think of the word external. Proprioceptors. These neurons are carrying information to the brain that is relevant to your body position in space and how you're moving. Okay, and then finally, the think of internal. Okay, so this is carrying internal information related to, say, pain, pressure, remember those stretch receptors, and oddly enough, taste, because we even have some taste receptors going all the way, not just from the mouth, even down into the back of the tongue towards the pharynx. Okay, the motor or efferent neurons. Okay, remember these are coming from the central nervous system. And I said they're all going to be the multipolar neurons. Okay, that last category of neuron. So we have somatic motor. Okay, so somatic motor neurons innervate your skeletal muscles, the voluntary muscles. If you want to scratch your nose, that's traveling along a somatic motor neuron. Okay, now what's a little different here is the visceral motor neuron. And the visceral motor neuron is where we're going to find our ganglia. Remember, the ganglia are the swellings that are found along peripheral nerves. Okay, so how does this work? With visceral motor neurons, you have a series of 2 neurons that carry impulses from the central nervous system to whatever they're going to stimulate. And that where they end up is called the effector. Okay, so the effector could be a gland, smooth muscle, cardiac muscle, right? Remember, we're not looking at skeletal muscle because that would be somatic. So let's say that this information is traveling to, say, smooth muscle of the small intestine, right? And by stimulating the smooth muscle in the small intestine, you're going to have peristalsis occur. Okay, you don't remember what that is, go back to digestion. Okay, so the impulse leaves the central nervous system and travels through the first neuron. Okay, and remember, these are all multipolar neurons. Okay, this first multipolar neuron has its soma or cell body safely hidden within the central nervous system. Okay, so most of the neuron is outside the central nervous system, but the soma is safely protected within it. Most likely this is going to be a spinal nerve. Okay, so this first axon that leaves the central nervous system is called the preganglionic fiber. So remember, an axon can also be called a nerve fiber, so that's where that comes from. All right, now then there's going to be a synapse between the end of this axon and the next neuron, the next multipolar neuron. Okay, and this is going to be housed, it's going to have its soma inside the ganglia. And remember what surrounds that soma? Those are the satellite cells. Okay, and then a long axon from this second neuron called a postganglionic fiber. All right, there's a lot going on here. Okay, before we do another visual, I haven't really talked about the connection between sensory and motor neurons. I keep saying sensory goes in, motor goes out, but what about in the central nervous system itself? So the neurons that connect the sensory and the motor portions are referred to as interneurons. Okay, all the interneurons are in the central nervous system. Okay, they are multipolar. Remember, that is the most common type of neuron in the central nervous system. And these guys connect up the sensory and the motor neurons. And if you counted every neuron in the body, you would find that the majority of them are in this category. They are interneurons. All right, here's a great visual for you to study. Let me walk you through it. Okay, remember, we started looking at the different types of receptors. Interoceptors, they tell you what's going on in the body. So let's say you have pain in your gut. Okay, that's going to be carried via an interoceptor, okay, from the pain receptor in your gut. Okay, and this red color indicates that we're looking at a visceral portion. Okay, so this is visceral, and because it's carrying information in, it's sensory. Okay, so from the pain receptor in your gut via this visceral sensory neuron into the central nervous system here. where it will synapse with an interneuron. Okay, this is another neuron. Remember, it's multipolar. Okay, and then that interneuron, and there may be one as shown here, there may be many. Okay, that depends where this information is going. But ultimately, from the central nervous system, there will be a synapse with the first of those two neurons that we identified as being part of the visceral motor portion. Okay, so remember this first neuron here, the preganglionic fiber, has its soma inside the central nervous system. Then there'll be a synapse between the two neurons. Then we're going to find the second soma here is going to be in a ganglia. And then the long axon, the postganglionic fiber. Okay, and that's going to end at an effector. So let's say we were having gut pain. And so maybe we're going to stimulate smooth muscles, and there's going to be contractions to move food along through the digestive system. Okay, that might be what happens as an outcome, for example. Okay, now exteroceptors are going to be picking up information outside the body. Let's say, for example, you put your finger on a hot stove. Okay, those are pain receptors, heat receptors, and that information will travel this time along. This is going to be somatic sensory, okay, into the central nervous system, synapsing with an interneuron. Now, notice this little shortcut here. Okay, there are what are called reflexes. And when you have reflexes in the body, it can bypass the interneurons, making for a much more rapid response by the somatic motor neurons, traveling to, say, a skeletal muscle. And that would be important in the case of something like, say, pain or heat. You don't want a slow response to that. So you're not going to involve a lot of interneurons in responses to pain or anything else that might damage the body. So usually, even if there is an interneuron involved, it'll be a pretty short path. You don't need to have the brain consciously considering what to do as you sit there with your flesh burning, right? You don't want to wait. So that's going to be a very quick turnaround through the central nervous system. Okay, now, of course, the interoceptor may end up traveling back. The response to that information may end up traveling back via the efferent motor, okay, the somatic motor here. So let's say you're feeling gut pain, but you know from your experience that this is a sign you've got, say, I don't know, diarrhea, let's be gross. Okay, so now you want your response to be skeletal muscle to walk you to the restroom. So in that case, this central nervous system may end up synapsing, here we go, with some of the somatic motor fibers, which are going to lead to contraction of your skeletal muscle so you can hurry up and run to the bathroom. Okay, so it's not just, excuse me, that sensory goes to sensory and, somatic goes to somatic and visceral goes to visceral. It doesn't have to be that way. All right, so take some time looking this over. There's a lot here. All right, now I mentioned before that it's important to keep the soma of a neuron protected because it is possible for axons to be repaired if damaged. And that's what I'm going to show you now. So if we're in the class, I would ask if anybody has ever had like a surgery or a deep cut. So what will happen often after surgery is that you'll realize the area around the incision has gone numb. Okay, so what has happened is with the scalpel, the surgeon cut through some of the axons. These are axons of the peripheral nervous system. And you can see that this one is myelinated. See the cells there? That's a Schwann cell. So this is what will happen if you cut through a myelinated axon. Okay, so here's our cut. This part of the axon is still attached to the soma. Okay, so it will remain alive. Here's our myelin. You can see the nucleus there of the Schwann cell. Okay, now everything that is distal is going to die. All of that axon is going to die because it's no longer attached to the neuron. And so here's our macrophages. Remember, these are cells of the immune system. They're going to come along and they're going to eat up all of this dead, dying, damaged tissue. Okay, so we're looking at damage done to an axon. The Schwann cells, which give you myelination, are going to survive. And what that does is it leaves a little tube of Schwann cells. And that tube acts to help guide the end of the axon. Okay, so this axon, which is still alive, is going to start growing very slowly. This takes a long time, but it's going to grow very slowly along this tube. And eventually, if you're lucky, it can reinnervate its original effector. Okay. Now, just give you a little bit more information on action potentials. Okay, so an action potential happens. We looked at this with the muscles. This should be repetition for you. When the plasma lemma, remember that's the outside, the plasma membrane of a neuron, or what we call a neurolemma, okay, becomes permeable to ions. And remember, an ion is a charged particle. So here, Na+, this is a sodium ion. It has a positive charge. And here we have potassium ions, and those have a negative charge. Okay, and so what we have is a buildup of sodium on the outside. When an action potential is initiated, the sodium ions move from high concentration to low. So they're going to cross the plasma lemma and enter into this neuron. And the reason they move, this is just diffusion. Okay, so the action potential starts when the ions are able to move across the plasma lemma. Okay, and that ability to enter or to move across the plasma lemma passes down the entire length of the neuron. Okay, and remember that's called depolarization. So this is a polarized cell because now we've pumped all these sodium ions outside. We've made a charge around the cell. When the action potential travels, the neuron will become depolarized. So in order to do it again, you're going to have to, remember, repolarize the cell. And that's when we use ATP. So the more sodium ions cross, the more ATP we have to use to repolarize the cell, which helps to explain why myelinated cells use less energy, because there's fewer areas where the sodium ions will cross, because a lot of the outside of this neuron is now protected by that fatty coat of myelin. Okay, so it is important to know that once any kind of action potential starts, it's going to be propagated. It's going to pass along the entire axon. I said the same thing with muscles. And when we looked at action potentials there, it's not a little bit. You either have an action potential traveling along a neuron, or you have no action potential at all. Okay, so the sodium ions that I showed you, they rush in. And the potassium ions, we didn't get that far, but they're going to exit the neuron. Okay, so this is depolarization, the movement of all these ions. In order to repolarize the neuron, to do it again, you have to use ATP. Okay, you'll get a lot more details on these processes later, but I just want you to understand the basic idea of an action potential and why it uses energy. Okay, so how are these action potentials passed from one cell to another? Okay, we've gone over this before. This is our vesicular synapse. So here we have the end of one axon. Over here is the effector. Now in this case, it says it's a neuron. Remember, this could be smooth muscle, could be skeletal muscle, could be a gland, okay? But the neurotransmitter here inside these vesicles. This again indicates acetylcholine, which is the one we talked about before. In physiology, you will learn about additional types of neurotransmitters. But these neurotransmitters are dumped into this cleft, the synaptic cleft, where they quickly diffuse across the space and they bind to receptors in the second cell. When they do, that causes changes in the plasma lemma and allows for the ions to start crossing the plasma lemma, right? So now we're going to see again depolarization of this second cell. There is another kind of synapse. And if you go way back in your brain and in your notes, you will find that we talked about how one cell is attached to the cell next to it. And we did it when we were talking about epithelial tissues. Okay, so one of the ways I said that two neighboring cells can interact with one another is these molecules here. These are proteins. Each one of these is a protein. So there is a green protein here. Here's another one here. And these are called connexons. And so when the connexons bind together, they make a passageway or a channel that goes from one cell to the next. Okay, so what can happen with these synapses is here's our sodium ions that have entered the presynaptic cell. So this is the cell that's already had an action potential travel down its length. The sodium ions go through these channels, and as they flood into the second cell, this postsynaptic cell, it creates a new action potential. Okay, so in this case, we have an action potential traveling cell to cell without a neurotransmitter. Okay, that's important. This kind of synapse can be referred to as non-vesicular, meaning there's no vesicle, there's no neurotransmitter, or an electrical synapse. But where you're going to find it is smooth muscle to smooth muscle, or you can see it with cardiac muscles as well. And this means that as this muscle contracts, that contraction impulse, that action potential is going to pass very quickly to the next cell. So it gives you a rhythmic contraction of muscle. Okay, so these plasma membranes, here's one here, here's one here, they're bound by these connexons that form a channel, an opening from one cell to the next. All right, so we're going to stop here. I will be doing some additional nervous system later. Okay, but I do want to remind you this lecture will be covered on your final exam and not on exam 3. Okay, I will talk to you soon. Happy studying, and let me know if you need any help. Thank you. Take care of yourselves. Bye.